One of the most important parts of the eye is the cornea. The cornea is built up out of 5 distinct layers. From the outside to the inside of the eye, these layers are the epithelium, the Bowman's membrane, the stroma, the Decimus membrane, and the endothelium. The endothelium is a cellular layer that exists out of a monolayer of hexagonal cells. These cells maintain the hydration of the eye, which is important for the cornea to remain transparent. These cells do not regenerate, and thus their number slowly decreases when a person ages. At a certain point, enough cells are lost so that it is no longer possible to maintain the hydration of the cornea, and the cornea loses its transparency. The result is a disease known as Fuchs endothelial corneal dystrophy, which eventually results in blindness. Currently, the only way to tackle this disease is by means of a corneal transplantation, where the Decimus membrane with the endothelium are transplanted from cadaveric donor corneas to the patient. Thereby, the rolled up membrane is inserted in the eye through an incision. It is then oriented by tapping on the eye. The membrane is then unscrolled by using an air bubble. The air bubble is removed and a new air bubble is inserted underneath the membrane to fix it to the top of the cornea. However, there is only one donor available for every 70 patients. There is thus a huge donor shortage. To tackle this donor shortage, we are developing an artificial Decimus membrane that will act as a carrier for lab-grown corneal endothelial cells to replace the functionality of the lost cells. The membrane is produced in 3 different layers. First, a sacrificial gelatin layer is attached to a glass slide. This layer can later be removed to detach the membrane from the glass slide. Second, a layer of a biodegradable bioplastic is coated on top of the sacrificial gelatin layer. This layer will ensure the structural integrity of the membrane. The final layer exists out of a gelatin derivative and will act as a compatibility layer between the membrane and the cells. Once the membrane is produced, it can be seeded with cells. To separate the membrane from the glass slide, the membrane is incubated in warm water. This will dissolve the sacrificial gelatin layer, effectively separating the membrane from the glass slide. The membranes that are obtained in this way can then be used as an artificial implant to replace the lost corneal endothelial cells, causing patients to regain vision. It is important that these membranes are transparent. We were successful in producing membranes that were sufficiently transparent, as can be seen in this picture. Another important requirement is that membranes are able to sustain corneal endothelial cells. Also here our membranes proved to be successful. In the top left movie, you can see that the cells are able to divide and multiply on our membrane, and the pictures show that the cells are healthy and have the correct hexagonal shape.